to think about sexual reproduction. In animal cells, sexual reproduction happens through the process of meiosis. Your body has two different types of cells. It has somatic cells, or body cells, and it has sex cells. Sex cells make eggs and sperm. All of the cells in your body contain a specific number of chromosomes. For example, as I mentioned earlier, human beings have 46 chromosomes. Homologous pairs of chromosomes contain the same basic information. This is because all chromosomes carry genes that are located in specific locations on the chromosome's body, called a locus. Most chromosomes are autosomes. These are chromosomes that are not associated with determining your sex. The only exception would be the X and Y in humans. Those would be sex chromosomes. Cells that have two sets of chromosomes are called diploid. Gametes, like eggs and sperm, are haploid cells. That means that they only have a single set of chromosomes. Some species, particularly plants, can have even more copies, as many as 5 or 6. But for our purposes, we will be spending time thinking about diploid cells and haploid cells. So one of the big questions is, how do we go from a diploid to a haploid cell, and why would we need to do that? The answer to that question is meiosis. Meiosis is the process of producing non-identical haploid cells from a diploid parent. This is how we make eggs and sperm. On the surface, the phases of meiosis are very similar to what they are in mitosis, and they have very similar names. One of the things that we're going to be keeping track of in this particular set, though, is how many chromosomes each cell has. We're going to use a shorthand for that. Down at the bottom, you'll see that it says 2n equals 6. This means that this cell is diploid, 2n, and that the total number of chromosomes that it has is 6. This means that it has 3 homologous pairs. The first phase is interphase, just like in mitosis, and the cell looks pretty much the same. The chromosomes are all decondensed, they're inside the nuclear envelope, and the centrosomes have not yet started to grow. In the beginning of prophase 1, the first round of meiosis, instead of just condensing, the chromosomes are going to find their homologous pair. These pairs are then going to hold hands, essentially. They're going to form little crossover events that are going to hold the pair together. This forms what we call a tetrad. A tetrad is a connected pair of homologous chromosomes. The spindle will also start to grow, the same way that it did during prophase of mitosis. During metaphase 1, the chromosomes will line up, but this time, instead of lining up single file, they'll line up as pairs because they are attached to one another as tetrads. Instead of having the centromere separate during anaphase 1, the tetrads will break apart, and each whole chromosome is going to migrate to its side of the cell. Then there will be a brief phase in telophase and cytokinesis where the cell will go ahead and divide. At this point, there is now only one copy of each chromosome in the two daughter cells. This means that instead of being 2n equals 6, we are now 1n equals 3. That makes these cells haploid. This is sometimes confusing for students because they look at it and say, well, but there are 2 sister chromatids. Doesn't that mean that there are still 2 copies of every chromosome? It could mean that, except we're missing the other copy. We only have the maternal or the paternal copy, not both. So it's a good idea to count the number of chromosomes based on the number of centromeres that are present. In this case, each of these has only one centromere, so it counts as only one chromosome. Because we've gone from diploid to haploid, this is called the reduction step. Now we need to make sure that we have the correct number of sister chromatids per cell. So we're going to go through all of this division again. We're going to go through a new prophase where the spindle will start to regrow and the nuclear envelope, which was briefly present during telophase and cytokinesis, will break down again. This time, when we enter metaphase, the chromosomes will line up single file the way they did in mitosis. The centromere will break down, allowing the sister chromatids to separate in anaphase 2. And in telophase 2 and cytokinesis, the cells will separate again. Notice here that instead of having 2 diploid daughter cells, we have 4 haploid daughter cells at the end of meiosis. The reason why we have to do this is pretty simple. In order to be a human being, you have to have 46 chromosomes, and you have to have the right combination of them. If your cells were to just fuse together, you would take 46 chromosomes and another 46 chromosomes, and suddenly you'd have 92. I don't know what that would be, but it wouldn't be human. So instead, if we want to maintain 46 chromosomes, and we want to be able to have an egg and a sperm come together, we need to make sure that they only give half as many chromosomes. So the process is going to constantly go between you, the adult diploid organism, which has 46 chromosomes, to your sex cells, eggs or sperm, which are going to have half as many and be haploid, so that when they fuse, you get 46 again. All right, so let's make some comparisons between mitosis and meiosis. First,
Ripley,
both mitosis and meiosis
right
begin with diploid parent cells that have chromosomes that have replicated during interphase. At the end, mitosis is going to produce 2 genetically identical diploid somatic daughter cells. This means that they will have 2 copies of every chromosome, and that those copies will be identical between the cells, and that these cells are body cells. That's why they're somatic. During meiosis, we're instead going to produce 4 genetically unique haploid gametes in the forms of egg or sperm. So if we were to start with a parent cell that's 2n equals 4, that means that it has 2 homologous pairs. The first step is going to be prophase. In prophase, chromosomes are going to condense and the mitotic spindle will start to grow. In mitosis, the chromosomes will not associate with each other, but in meiosis 1, the chromosomes will find their homologous pair and form a tetrad. In metaphase, chromosomes will line up on the metaphase plate. In mitosis, they will line up single file, but in meiosis metaphase 1, they will line up in pairs because they are forming a tetrad. The process of anaphase, telophase, and cytokinesis are pretty much the same. In anaphase, chromosomes will separate, in telophase they will reach their ends, and in cytokinesis the cell will divide. In mitosis, anaphase will happen when the centromere breaks down, allowing sister chromatids to separate. But in anaphase 1 of meiosis, because chromosomes are tetrads, instead the homologous pairs will separate. Sister chromatids will stay together. Mitosis is done after 1 round of cell division and results in 2 daughter cells that are identical to one another and are diploid. 2n equals 4. However, meiosis needs to go through a second round of cell division called meiosis 2. Meiosis 2 looks almost exactly like mitosis and goes through the same phases: prophase, metaphase, anaphase, and telophase. Unlike in metaphase 1, in metaphase 2, the chromosomes will line up single file, and this will allow sister chromatids to separate. At the end of this second round of cell division, we will be left with 4 unique haploid daughter cells. Alright, so now that we've talked about the 2 types of cell division, asexual cell division in mitosis and sexual cell division in meiosis, how do we get from all of that to this? Where does variation come from, and how does sexual reproduction help that happen? Well, there are a couple of places that this can come from. The first one is something that we call independent assortment. This relies on the idea that every time chromosomes line up in the form of a tetrad, they could line up in any way. For example, we might have a situation where all of the blue chromosomes end up in the same daughter cell, whereas in another situation, some of the blue chromosomes and some of the red chromosomes might be mixed up. The more chromosomes you have, the more possible combinations you can get. There's a formula that we can use for this, which is 2 to the n, where n is the number of homologous pairs. So you can see pretty quickly that when you have as many chromosomes as you do, 23 pairs, that already gets us to more than 8 million possible combinations just for making eggs and sperm for a single person. And of course, we have to add an egg and a sperm together. So, actually, the likelihood of any possible outcome is 1 in 64 trillion. Now, I've heard people say you're 1 in a million before, but I guess they were underestimating. So why does this matter? Well, it has to do with the differences between homologous chromosomes. So I told you that these chromosomes carry the same information in that they might decide what color your eyes are supposed to be, but they might have different ideas about what that color is. Maybe one of them says green and the other one says brown. This means that there are different possibilities for what each egg cell or sperm cell might convey to offspring. Let's think about this in the context of mice. Let's say that there is a chromosome that has instructions about 2 different things. The first thing it has instructions for is what color your coat is going to be, whether or not your coat is brown or white. And another location on the chromosome determines what color your eyes are going to be, whether or not they are black or pink. We talked about how homologous chromosomes make tetrads during meiosis. This means that during meiosis 1 of the chromosomes will go into one cell and the other homologous chromosome will go into another. Now, in our example here, the brown and black are on the same chromosome, and the white and pink are on the homologous chromosome. This means that when they separate, they will either give the information for black and brown together or the information for white and pink together. Which one you get is just going to be determined by chance. Now, to make things a little more complicated, there's another way that we can increase our variation, and that's called crossing over. Crossing over is an exchange of corresponding segments between non-sister chromatids. This happens because of the way tetrads form. When a tetrad forms, there are chiasma, or crossover sites, that help hold the tetrad together. Remember, we had talked about them kind of holding hands? When the chromosomes are pulled apart during meiosis, this can sometimes break the chiasma, exchanging parts of the chromosomes with each other. So how does that lead to variation? Let's think about our mouse experiment. The red chromosome contains information for dark coat and dark eyes, whereas the blue chromosome contains information for white coat and red eyes. If a crossover event happens between these 2 genes, then they might be exchanged. This would result in 4 possible different combinations. 2 that look like the original parents, dark with dark eyes, light with pink eyes, and 2 that are recombinant or in between. For example, the second mouse has a dark coat and red eyes, whereas the third mouse has a white coat and black eyes. Something as simple as where chromosomes end up through independent assortment, or whether or not a crossover event happens, like in this mouse example, increases the amount of